Hello students, welcome to the biology class. Today we are going to continue with our very important chapter, photosynthesis in higher plant. And today we are going to discuss the first mechanism of photosynthesis, that is your light reaction or photochemical phase. Light reaction or photochemical phase involves the following processes or steps. First is light absorption by the chlorophyll. Number 2 is water splitting in the presence of sunlight known as photolysis. Oxygen released as a byproduct, and at the end of light reaction, formation of ATP and NADPH takes place. ATP and NADPH, they are the high energy chemical intermediates. So here in this diagram you can see the light that is coming from the sun as the ultimate source of energy falls on the leaf, which is green in color, and that light energy is being absorbed by the chlorophyll. That is your first step will be there, that is light absorption. Then there is a water which will be splitting out into hydrogen ions and oxygen is liberated out as a byproduct, and NADPH and ATP, they are being synthesized, which will be used by the dark reaction for the synthesis of sugar with the help of CO2 or with the reduction of CO2. So one by one we will discuss the steps which involve in the light reaction. So this is the Z scheme of light reaction, in which this is your LHC, means your light harvesting system, where the light is going to be fall in the form of photons that is absorbed by your photosystems 1 and 2. Electrons will be emitted, accepted by the electron transport chain, and in between there is a synthesis of NADH. Then there is a second photosystem is there, which will get excited, electrons are emitted and accepted by the primary acceptor, and then they will synthesize NADPH. So when the PS2, that is your photosystem 2, absorb the red light of 680 nanometer wavelength, electrons are excited and transferred to the electron acceptor. The electron acceptor passes them to the chain of electron transport system consists of cytochromes. This movement of electron is downhill, that is from upper to the lower side, in the terms of redox potential scale. The electrons are transferred to the pigment of PS1, that is your photosystem 1. Simultaneously, the electrons in PS1 are excited when they receive the light, that is the red light of 700 nanometer, and transferred to the another electron acceptor having a greater redox potential. These electrons move downhill to the molecule of NADP. As a result, NADP is reduced to NADPH. Transfer of electrons from PS2 to PS1 and finally downhill to NADP to NADPH is a Z scheme. So you can see that is the shape of that scheme, that is from LHPS2 to electron acceptors, then to the electron transport system, then PS1 to the electron transport system, then downhill to NADPH. That is in the Z shape, that is due to the zigzag shape. This shape is formed when all the carriers are placed in the sequence on the redox potential scale. So here the electron transport system, as we have discussed in our respiration, They are placed as they are having the redox potential scale. So the next step, that is your first step of your light reaction, that is splitting of water, that is known as the photolysis. The water splitting complex in PS2 is located on the inner side of thylakoid membrane, and when the water is split into hydrogen ion, into electrons and oxygen. So PS2 can supply electron continuously by replacing the electrons from the water splitting. Thus PS2 provide electrons needed to replace those are removed from PS1. The protons are used to reduce NADP to NADPH, and this oxygen is liberated as byproduct of photosynthesis. The next is the photophosphorylation, that is the synthesis of ATP by the cells in mitochondria and chloroplast. This is called as the photophosphorylation. In mitochondria, we did in our respiration chapter, that is called as the oxidative phosphorylation, and when that is synthesis takes place in the chloroplast in the presence of sunlight, then it is being called as the photophosphorylation. Photophosphorylation is the synthesis of ATP from ADP in chloroplast in presence of light. It occurs in two ways. Number one is non-cyclic and another is cyclic. So first we will discuss the non-cyclic photophosphorylation. It occurs when two photosystems work in series. First, that is PS2 followed by PS1. Fine, the first is PS2 and then that is PS1 through an electron system chain as seen in the Z scheme. Here ATP and NADPH are synthesized. It is non-cyclic process because the electron lost by the PS2 do not come back to the PS2, that goes to the PS1 and then to the NADP to form the NADPH. That's why it is known as the non-cyclic photophosphorylation. So next is the cyclic photophosphorylation. It occurs in the stroma lamellae when only PS1, which is of P700, is functional. The electron is circulated within the photosystem and ATP synthesis occurs due to the cyclic flow of electrons. The lamellae of grana have PS1 and PS2. The stroma lamellae membrane, they lack PS2 and NADP reductase. The electron does not pass on to the NADP, but it is cycled back to the PS1 through the electron transport system. Here only ATP is synthesized and no NADPH is reduced. Cyclic photophosphorylation also occurs when only the light of wavelength beyond 680 nanometer are available, because they function at 700, and if the light is available beyond the 680, only then it will function individually. It occurs in the stroma of lamellae, whereas the non-cyclic that occurs in the stroma, and they are having grana as lamellae of grana, and this occurs in the stroma lamellae where only the PS1 is there. The electrons which are being emitted by this PS1 receives by the electron acceptor, then they passes to the electron transport system where the ATP is synthesized and they are back to the PS1. No, you can say the formation of NADPH, as the electron does not pass to NADP, but they are cycled back to the PS1 through the electron transport system or the electron transport chain. So these are the two pathways by which the ATP or NADPH is synthesized. One is non-cyclic, in which both the systems are involved, and another is a cyclic in which only PS1 is involved. In non-cyclic, we have a synthesis of ATP and NADPH, whereas in the cyclic, there is only the synthesis of ATP and no synthesis of NADPH. Next is light reaction phase is chemoosmotic hypothesis. It explains the mechanism of ATP synthesis in the chloroplast, as we have discussed in the respiration too. So chemoosmosis, the movement of the ions across the semi-permeable membrane, it occurs in the chloroplast as well as in the mitochondria. Chemoosmosis needs a membrane, a proton pump, proton gradient across the thylakoid membrane. So this is the thylakoid and this is the membrane, and across this the exchange of the protons takes place is known as the chemoosmotic hypothesis, which helps in the formation of ATP. Splitting of water takes place on the inner side of the membrane. So the protons accumulate in the lumen of the thylakoid. This is the lumen of thylakoid. Photolysis takes place inside this so that the gradient or the concentration of proton is more in the lumen as compared to the membrane. The membrane, they are having the electron transport system. As the electron moves through the photosystem, protons are transported across the membrane. It is due to the removal of protons from stroma of the following reason. The primary electron acceptor is located towards the outer side of the membrane and transfer its electron to the hydrogen carrier. So this molecule removes the proton from stroma while transporting the electron. So this is your grana and this outer surface is known as your stroma. When this molecule passes on to the electrons to the electron carrier on the inner side of the membrane, so this is your electron carrier, proton is released into the lumen of the membrane. As the electron moves through the photosystem, protons are transported across the membrane. It is due to the removal of protons from stroma for the following reason. The second reason is NADP reductase enzyme is located on the stroma side, that is outside. This is your NADP reductase enzyme. Along with the electrons coming from the PS1, protons are necessary to reduce NADP to NADPH. These protons are also removed from the stroma. Hence, the protons in the stroma are decreased, but in the lumen, the accumulated protons are increased. It creates a proton gradient, means the difference across the thylakoid membrane and decrease in the pH in the lumen. Breakdown of the proton gradients leads to the synthesis of ATP by the enzyme ATP synthetase. So ATP synthetase consists of two parts, that is CFO, it is chloroplast FO. This is the part you can see, that is the head and the stalk portion. It is embedded in the membrane and form the transmembrane channel. It carries out the facilitated diffusion of protons across the membrane to the stroma. It results in the breakdown of the proton gradient. And F1, it protrudes on the outer surface of a thylakoid membrane. So they are protruding out from the outer surface of the thylakoid membrane. The energy due to the breakdown of gradient cause the conformational change in CF1 particle. It makes the enzyme to synthesize the ATP. Energy is used to pump protons across the membrane to create a gradient or the high concentration of protons within the thylakoid lumen. ATP synthetase has a channel for the diffusion of protons back across the membrane. This releases energy to activate ATP synthetase that that catalyze the formation of ATP. With this, we finish our first mechanism, that is your light phase, in which we have synthesizes the ATP, NADPH. Along with that, oxygen is liberated, and we have discussed about the chemoosmotic, that how the NADPH is helpful in forming the ATP. In our next lesson, we will discuss the dark reaction, that how the carbon dioxide is reduced to synthesize the carbohydrate, that is the sugar. Till then, stay home, stay happy, and have a nice day. Thank you.